thank you for the kind opportunity also to talk to you today about some of the work we are doing at Maastricht University within the Merlin Institute for Technology-Inspired Regenerative Medicine. So what is actually regenerative medicine? This is actually a relatively new interdisciplinary field that is actually aiming at regenerating tissues in our body that have lost the capacity to do that, either because of traumatic events, as you can see here on the upper left corner of probably the Benjamin Button of soccer or football, or because of aging and the fact that with increasing the quality of life, we grow older, and by growing older, we simply use our biological tissues a lot more than in the past. Normally, this is done when a tissue, like for example, our joints or our bones are worn apart. They are replaced by synthetic artificial implants, as you can see here on the right picture, which are very far from what typically our native tissues are, in this case, bones. If we replace bones with metals, we can in part recover the mechanical functionality of our joints, but certainly we cannot recover the biological functionality. And this results in a number of different complications and pain, multiple surgeries, and oftentimes also scarcity of biomaterials to perform the job. In other kind of parts of our body, for example, for our blood vessels or our heart valves, or as you see on the bottom, for our eyes, we again have so far different kind of replacements of the body that again allows for a temporary solution that anyhow will have to be replaced in the near future of these patients. And so what actually we would like to do with regenerative medicine is actually also what some of the other animal species, such as salamanders, can actually do quite well by themselves. You may have been already seeing this a number of times, but if you would subject a salamander to an amputation, the salamander actually would be able to regenerate its own limb by coaxing actually all the biological processes that are needed for the limb to regrow. Somehow, actually, as humans, we have lost this pretty much early in time after actually we have been born from our mothers. So what actually aims regenerative medicine to do is to use actually either cells or materials, or cells, materials, and these little colored worms, if you wish, here on the bottom right, which identify cocktails of biological factors separately or together, to then really restore not only the mechanical functionality, but also the biological functionality of the tissue that is damaged or diseased. If we look a little bit more closer to how this kind of materials can be designed, a branch of regenerative medicine called Biofabrication actually really aims at specifically determining space and time how the materials and the cells and eventually the biological factors can be placed together a priori. What kind of materials we use for different kind of body parts? Actually, we have different kind of materials. In the case of bones, considering that these are a mixture of collagens and hydroxyapatite, which is a ceramic. Oftentimes we do look at ceramics. Now we are not going to implant Delft pottery ceramic, but that's just to give you an idea of the kind of similarities of where you find also this ceramic material in daily life. Similarly to when we look at softer tissues such as heart valves, these typically are made by a very intricate matrix or mesh of fibers, which are made by polymers or plastics, unless our cotton shirts or other kind of textiles need to be biodegradable. And doesn't really matter if they are very colorful, as you see here in this image, but the most important is that they have to degrade away in time as the tissue is being replaced. And so with this kind of different materials, we can also create all sorts of different mathematical models that predicts also their kind of properties. Here you see that, for example, if we use a specific set of fabrication technologies called 3D printing, we can choose, again, a priori, the kind of material that is chosen, here represented by a specific physical parameter called mesh size, the porosity of this kind of porous textile structures, and by interpolating these two parameters, for example, we can modulate the stiffness or the elasticity of the final construct. We can then use materials that are already known to regulatory agencies, so that typically they would have a higher acceptancy when moving from the bench to the bedside. In this case, for example, we have used a material that specifically resorbs almost completely 90% of its own weight after 1 year from implantation, and has similar stiffness as the cartilage, which is what we have on the upper surface of our joints. And it allows us to essentially move without any friction. And again, this kind of material, for example, has been already evaluated for other kind of medical device purposes by regulatory agencies such as the Food and Drug Administration in USA. So we have then placed different kind of cells on these kind of scaffolds. And again, I'm going to make a very big jump in terms of scientific data, because in the context of 7 years, then we were able to then see which kind of cocktails of adult stem cells and specialized cells of the skeletal tissue, in this case chondrocytes, which are the cells of cartilage, were able to generate the best articular cartilage, both in vitro as well as in animal preclinical models. And this allowed us then to move to a clinical setting where now about 300 people, or a bit more actually, are walking through these kind of implants, showing not only a regenerated cartilage, which is pretty much the same as the native cartilage after 1 year from the implantation, but also an increase of what you see here on the left side, this COS scores, which is a semi-quantitative test to measure the recovery of quality of life of these patients. That's actually, this goes relatively well for a number of tissues in our body when we target only damage that is specific to a single tissue. When we look at multiple tissues together, we have to go back to the drawing board, and that's not something where regenerative medicine has been able yet to deliver so many products already to the clinic. And so that's also where biofabrication could play a better role. What is actually this biofabrication? Essentially, you may have also heard that this often called as bioprinting. Bioprinting is one of the different techniques that biofabrication, within the realm of biofabrication. Essentially, this is a field where either we direct in space the arrangement of cells, materials, and biological factors, and in order to recreate a tissue, or that we use a sort of mini blocks, if you wish, sort of biological Lego blocks. And then, like when we were a child, by putting one Lego block on top of each other, we can recreate a final building. In this case, not a building of our imagination, but a building that would serve the purpose of restoring the biological function of our body. Now, to give you a simple example of how this could be done in a smart way, when we think about many tissues in our body, these are typically also subjected to specific mechanical loading. This could be compression or tension most of the time, or a combination of the two modalities. And then sometimes actually when we apply ultrasounds on biomaterials that are being the house of our cells, we also can create a mechanical loading in a programmed manner. This allows them to change specifically the architecture, the skeleton of which is internal to the cell membrane, which specifically for stem cells allows them to influence their differentiation state. So we have actually done this a little bit. We have, you see here a little bit a mocking of the scientific question that we have asked ourselves. So do bone cells like more classic or rock music? Essentially what we have thought was to use ultrasounds at different amplitudes and frequencies to see if this would result in different nano vibrations that would give some sort of mechanical loading kicks to the cells and would result in increase of their bone forming capacity. So this actually was the work of a former postdoc in the lab who is now a group leader at the University of Basque Countries in San Sebastian. Sandra actually was very much interested in creating scaffolds that could actuate under an external stimuli. And so she actually did that by using two very common biodegradable polymers. I will not go too much into the chemistry for the purpose of brevity. But essentially what she did was to create some sort of Janus scaffolds. So scaffolds that actually would have one polymer on one facade and a completely different polymer on the other facade. And then by actually subjecting these scaffolds to an ultrasound wave field, it would indeed control the modulation of the deformation of the vibration that these scaffolds would be subjected to in the order of a few hundreds of nanometers. Would that result in a better bone formation? So it seems that indeed for a specific frequency of the wavelength of the ultrasound wave field at 40 kilohertz, there were a number of different proteins that were better expressed and secreted by the cells in these Janus scaffolds, such as fibronectin or collagen type 1, which are very much typical of bone, as well as a number of different genes, again, typical for bone differentiation of stem cells, were also more highly expressed than the non-Janus conditions or the non-stimulated conditions. Now in bone, again, as I said before, it's not only about collagen, but it's also about the mineral component, so the ceramic component. And again, here, what we have seen is that indeed with these Janus fibers with ultrasound stimulation, we saw a more abundant amount of mineral formation. And yet again, I told you before that when we think about more complex structures in our body, things get easily complex. So if we also think about our bones, not only simply for their bone composition, but also for the blood vessels or the neurons or the neural network that make them working at equilibrium in our body, at homeostasis. Or if we think about organs such as a heart or a kidney, these actually would be much more complex to regenerate specifically with one combination of materials and cells only. In this case, actually, is where bioprinting could play a role. And again, here we have a number of different technologies that composes the bioprinting library. You see here some of the modalities. We started with first attempts by simply hijacking a former Hewlett-Packard or whatsoever printer, and then instead of having our ink droplets, having cells as our ink droplets, and from there we move to continuous extrusion of cells, either alone or together with a hydrogel carrying them on a specific final shape. We also nowadays use levitation mechanisms, either by magnetic or acoustic levitations, again, with the idea that in this case, we can better position not only single cells, but also cell clusters in space in a specific location. I'm not going to go for the purpose of time into examples here, but we are together with a number of younger PIs at the Institute working on different kind of applications, and in general, the whole bioprinting palette here at the Institute is led by Carlos Mota. who specializes also on kidney bioprinted models. With this, I'm at the end of the presentation. I'd like to introduce who I call my superheroes, taking inspiration from an old cover from Nature. It's a highly interdisciplinary set of researchers. You see here some of the faces, in the meantime also not complete, and here also some of the names. Thank you for your attention, and thanks also for the funding that makes this possible.
Thanks a lot. Silent claps and loud claps. Very fascinating talk. Are there questions? I don't know whether I see if there are any hands raised. You can also just unmute yourself and ask your question. I have one, and I hope you won't hate me for this, but I'm wondering, as someone who's really in a completely different field and not understanding a lot of what you're doing, what's sort of the holy grail of this research, would you say? I mean, what would be the hardest tissue to regenerate in the human body? And why, basically? Sorry, very basic.
No, fine. I mean, that's fine. I mean, I think if you look at the previous talk from Valeria, brain would certainly be quite a challenge. But maybe I'd stick to what I just mentioned at the end of the talk. I think kidneys are quite a tough cookie, because they are comprised typically by more than 26 specialized cells. And a very intricate composition of the proteins that the cells, that each of these cells will do to make our kidneys, filtering our bloods and separating, therefore, purified blood from urines. This indeed is quite a challenge. So in general, when it's about a single tissue, in many cases there are already things that have gone from fundamental research to the clinic. When it comes to multiple tissues together or to organs, this is still quite far-fetched.
Okay, interesting. Andrea's hand was up first.
Yes, so the picture of the different salamanders made me sort of think about, because you also mentioned that we as humans have lost the ability or lose the ability to regenerate when we are born, or maybe even before that, I'm not sure. But is there a biological benefit of not being able to regenerate, you'd say, your limbs?
I cannot think of one, but maybe there is one. But of course, I mean, there are also ways that you could substitute the function, especially in the case of limbs with prosthetic devices. But there are also quite a number of discomforts in, for example, how the sleeve that typically connects a prosthetic device to the amputated limb, that as you walk, actually there are a lot of frictions and a lot of problems connected, for example, to the climate. For example, if it's a very humid environment, there is a lot of slippage, and this also creates a lot of social discomfort for the patients. I mean, so you could argue that in principle, if you would have a better acceptance of prosthetic devices and you could solve these kind of interface problems between the artificial limb and the native limbs, you could eventually avoid to regenerate a biological tissue. But yeah,
is it very energy consuming? Because I'm thinking that if we were able to regenerate at some point, why couldn't we, like the salamanders, continue doing so? It must be, I guess it must be super complicated, and from a sort of perspective.
Yeah, for sure. There will be also indeed quite a lot of phenomena at the metabolic, from a metabolic point of view, to be a better understood also for humans. Yeah, for sure.
Thank you for a very nice talk.
Anna.
Just following on from that question, in terms of what do we actually regenerate, because our hair and nails still grow throughout our lifetimes. I wonder what's the sort of difference. I know the sort of, ectoderm, what's the sort of origins are different for hair and nail, but is there some lessons that we can learn to help and promote regeneration in some other tissues?
For sure, I think you can learn it from a physiologically self-regenerating body, right? I mean, indeed, when there is no damage or there is no aging of our body, certain parts of our body actually regenerate quite well, right? In the case of aging, also our hair at some point and nails stop regenerating themselves. In the case of traumatic disease, I mean, this typically can happen unless there is a more severe cause of the trauma. So if for example, if you think about burn victims, when you have a deep burn and of a large extension, what we can do now with this actually is not repairable at the moment with a native process. What we can do actually with regenerative medicine is to recreate the epithelial barrier, but we cannot yet recreate hairs and the sweat gland capacity, so that in a way there is a barrier for external pathogens, but there is not yet a barrier in terms of maintaining the self-regulation of the body temperature. So that's still quite a impairment for people that have suffered from a burn, from an extensive burn accident.